Okay, so microbes, I know that school children in schools, we don't study much about microbes, but we study them or not, they are all around us, and our survival actually depends on microbes. They are everywhere in soil, air, water, and also on our skin. So why does our survival depend upon us? Because microbes play a vital role in on the planet and making it a livable planet for us, and the only planet that we know can sustain us. So what they do is they do cause diseases, but at the same time they also are very important, and they are usually in textbooks known as decomposers. So when plants or animals die, they decay, and these decomposers act upon them, turning them into fossils or organic matter. So they form a vital link between what the resources that plants and animals utilize and then cycling them back to the environment. So as you see, this is called as biogeochemical cycle, where plants and producers and composers, consumers take up the useful elements from the environment, and decomposers help to cycle them back to the environment. And this biogeochemical cycles are important link to the climate changes that we see in today's environment. And sometimes microbes can have very essential answers to how we can combat climate change or hazards of climate change that we are facing today. So if you want to know more about biogeochemical cycles, there are a lot of articles, very useful articles on internet, and or you can ask me more questions about that later. Secondly, microbes are also very much part of our body, and they help us digest our food. Also, some vitamins that are very essential for our body growth are produced by microbes from the food that we eat. Our body cells do not produce vitamins. Microbes are therefore essential to produce these vitamins in our body. And you might have heard about gut microbiome. And yes, so gut microbiome are really essential for digestion and energy consumption of from the food, and passing them to the organelles or different organs of our body. And also, like plants and animals, they form a unique layer over them. So as you can see here, here is a plant root, and then there is a fluorescent bacteria, which is made fluorescent in order to visualize it properly. And as you can see, it forms a very hard layer around the root. And this itself tells us that it is present all over. And this is a useful bacteria, so it kind of combats or protects plants from pests and other pathogens that could be harmful for the plant. Likewise, even on our skin, they form a protective barrier, and with our immune system, they help to combat disease-causing agents. So in a laboratory or in a microbiology lab or research centers, they're usually observed under a microscope, or they are used and grown as these communities. And these communities are called as colonies. I'm going to refer to this colony elsewhere when I'm going to talk about one of the practical tasks that you can do in schools. So please remember, what is a colony? Colony is a community of microbes. So microbes are very microscopic, right? But these single cells, they divide and form these clusters. And therefore, you can see a visible colony that is grown on a solid surface. Apart from microbiology lab, you still observe microbes in our day-to-day life. Something like kitchen science, which is curdling of milk, something that is done in every Indian household, and something that is part of classical microbiology. So what is classical microbiology is just inoculating or introducing microbes to a new environment. And that is what we do when you make curd, right? You use previous curd in a new batch of milk and then allow it to set. And what you are using in form of previous curd is nothing but bacteria that then get introduced to the milk, and they start utilizing the lactose in the milk and form acids, and therefore you get curd. Likewise, you see sometimes that your bread or maybe even chapati gets a black mold on it. And after you see that, you just throw it away. But if you are a curious person, maybe you can take a magnifying glass next time and observe that what are these black structures and how are they formed. And closely observing them, you might see that they are hair-like structures, and they have some beautiful morphology. Of course, they are not very black. They are sometimes green or even sometimes white. And so this is something is what is called as bread mold, and it is a fungus. And there is also on our ponds, you see this mossy layer, and this mossy layer is formed by single cells that also have chlorophyll in them. Chlorophyll allows cells to utilize sunlight and make carbon out of it and give out oxygen, something that also plants do, and a process called as photosynthesis. And these are microscopic algae who do that. So now we saw that we have curd and we have bacteria in curd. You have bread mold, but also our garden mushrooms are part of fungus. But what are these other three types? So I already mentioned algae. Algae are like plants, but microscopic plants, because they have chlorophyll in them and can do photosynthesis. And protists are like microscopic animals. They eat other microscopic beings like bacteria and also other protists sometimes. And then you have your viruses that cause common cold, and something that we know that virus always causes disease. And so these are 5 main groups, how microorganisms are classified as. The main difference between these 5 are that, so what differentiates between living and non-living being is presence of genetic material. So we as living beings have genetic material. A stone does not have a genetic material. So a virus is a simplest form of living being where the genetic material is covered by an envelope. And that's it. That's how viruses are. Next, bacteria are a step further. A genetic material is not only covered by an envelope, but the envelope is also very rigid and has certain hair-like structures on them and also certain proteins on them that help them take up things from outside environment and give out things from inside environment of the cell. And these hair-like structures help them to swim in the environment that they occur in. So they can swim in your gut, they can swim in your water, they can swim in your milk. So that is what hair-like structure helps them do. Fungi, on the other hand, have well-defined structures. As you can see closely, a mushroom has a stalk or a stem, and then it has this upper body. Usually, if you see a bread mold, you will also see such structures on the bread mold. You will see hair-like structures, which are the stem or stalks, and then you have an upper body. This upper body contains spores, something called as spores. Spores are nothing but seeds. Plants have seeds. When seeds are go to a new environment, they germinate and form other plants, right? So fungi also have spores that are thrown in the air, and they form other environments or other surfaces to catch and germinate or grow on. Likewise, algae, as I said, they have chlorophyll inside them. They are minute microscopic cells, and protists are microscopic animals. Something that differentiates these two from the other simple microorganisms is that they also have other organelles like mitochondria, like just our cells. Our cells are also microscopic, but they are still a higher form of organism on this planet. So this is how we differentiate these 5 categories. Mostly you use microscopes to observe them, but for certain higher organisms, you can also use magnifying glass. So next time you see a microbe, you have to first see if you can see it from a microscope, for magnifying glass or not, like fungi or algae. you can easily see with with magnifying glass. Now this boy has chicken pox. What kind of microbe do you think causes chicken pox? Any ideas?
Guys, you can wait. Just type your answers in the chat box, or if you want.
I'm getting some answers, but I'll give you few seconds more.
Great. Most people are saying virus.
Okay, yeah. So most people are very correct. It is caused by virus.
Guys, please feel free to share your responses by unmuting yourself on the mic in case you're not able to chat on the chat.
Yeah, that will also be fine. So now the second question. Which group does amoeba belong to? Now, amoeba is an organism that we say that we all have evolved from amoeba. Which group does amoeba belong to?
That's protist, isn't it? Protista?
Yes.
Hello?
Yes, you're right.
Yeah, okay. Yeah.
Yeah, so protozoa is part of protists and amoeba is also part of protists, and amoeba belongs to the group protists. The third question, you want to bake a cake, what will you add? What organism will you add in the cake? To
Bacteria.
bake bacteria?
That's yeast.
No.
Fungi.
Yes,
yeast.
And yeast is what?
It's a fungi.
Correct. So you add yeast in order to bake your cake because it helps your cake to rise, and it belongs to group fungus. So how do you culture microbes? I know you come from different backgrounds of the schools, and I understand that the schools do not have sometimes grants or monies to sponsor certain techniques like microbiology techniques, which involve a Petri plate. This is called a Petri dish, and we have a semi-solid surface on it, just like a jelly, and you culture it on this normally. And after growing, it takes about a day or two for microbes to form these structures, like I said before, colonies. So what you can do in order to do this at school level, and the other reason why this is not also a practical thing to use at the school, because these are higher study techniques, and sometimes 8th graders are too small to actually deal with such colonies, but they can deal with friendly microbes like microbes around us, on our hand, on our mobile phone, on our pen, in soil. Wherever they want, they can find what microbes grow. And so the easiest way that you can do is take a jelly packet, and while making the jelly, put it in the container, a container that has a wider surface would be useful. Once the jelly is set, what you can do is you can take samples from soil, skin, mobile phone, some objects that you use normally and you're interested in. There was an interesting question that someone asked, that what are the microbes that are in landfills? So maybe if you're curious, you can also get a sample from there. And these samples are there out in your environment. So I don't think they are harmful. They are very harmful for children to even use. And give it a day or two, and you will see these beautiful structures that develop. And maybe this is something that you can do at school level to introduce microbes to children.
How do you transfer these samples from different sources, soil or your skin? How do you transfer them onto the jelly plate if they're doing it in school?
Okay, yeah, very good question. So normally what I would do to transfer soil, I would just maybe put soil and dissolve it in water, and then take some drops and put it on the jelly for it to grow, and that is enough. When you have objects, I think the objects are best stamped. So if it is a hand, and a jelly, I can just stamp the hand on the jelly, just like this person must have done to get this this nice pattern of hands and hand microbes that are there on, probably are growing on his hand at that particular instant. So this is how you can introduce it on the jelly. So it's basically just putting microbes or introducing microbes to this particular jelly kind of Petri plate or a dish that you have created. And you can take samples from anywhere. You can take water samples, for example, if you want to see if your drinking water has some really bad looking microbes, you can do that as well. I know it is a very bit risky. Maybe you won't want to drink that water later. So it is up to teachers to decide what samples to find things from. But I think hands and the usual objects that we use are very interesting objects, and these are microbes that live with us. They are our microbiome, so they are not harmful. It's only that it will be possible for you to see them in form of these, and sometimes it might make people very aware, or people feel very dirty about things. And it is also another good way to introduce sanitation in schools, like why you need to wash your hands before eating or before doing some important things, and why cleanliness is important.
So I'm really interested in doing the jelly in a container experiment in my classroom.
Yeah.
One of the questions I had was when we grow microbes in a laboratory, we grow it on media like agar. It has a blend of nutrition.
So Winogradsky column is another, it's nothing but a replica of a pond system. What happens in pond, you get it and set it up in your in a place in your school backyard, or even in your house sometimes. So it's nothing but a cylindrical structure where you fill it with mud so that you have a difference in oxygen and also difference in sulfate. I'll briefly go through it all over again with this link. So what you do is you just take a cylinder surface, fill it with mud, and then with any kind of water. You can use pond water or rain water, or even your drinking reservoir water. So this is a link that is attached to the presentation. So when you have this presentation, you can use this link to go through this particular website. It is called Make a Home for Microbe, and this is called a Winogradsky column. And here children, same like 7 or 8 graders, or even smaller, are doing this technique where they take a cylindrical bottle. You can use the soft drink bottle, or sometimes you can also use glass bottle, and you can mark. There are 2 markings that they do. Okay, so one marking is for a basal layer. Normally your basal layer should contain cellulose and carbonates and sulfates, and they come from eggshells, paper, and lots of mud, this thick mud. And so that's what they are doing. They are using paper and eggshells and making this first basal layer. Next, they are using normal garden soil to add up to the second layer, second mark that they made. And then they are using garden pond water, or you can also use rain water. It depends on which kind of water you use, what kind of colors you see in this column. And once you add the water, you close this particular container with a muslin cloth in order to allow the aeration. So this is how the column looks. You can leave it next to the window for 8 to 10 weeks, sometimes even for months, like 6 months or so, to observe colors that happen in this particular container. So what happens here is, the way it is formed, you have air coming from the top, and therefore as you go bottom, the oxygen goes on decreasing, whereas sulfate concentration goes on increasing as you go towards bottom. The best thing about this site is that you can click on these blue dots and you can know what bacteria, what kind of bacteria are grown in each layer. So this site is really useful for that. You can know what is what in each layer. And this is how you can make a Winogradsky column. So I just wanted to say that after 8 to 10 weeks, this is how your bottles or Winogradsky column would look. Depending on what is your water source, what is your myrrh source, there are different kinds of colors that you can see. So this activity can be done in groups, and each group takes myrrh and water source from different locations. And then you can see what are the different colors that are formed. And this is also kind of replica of a pond. Of course, in our ponds and lakes, we don't see them see these enhanced colors. But then there is a lake in Yellowstone National Park where you can because it has higher sulfur, you can actually see these colors in broad daylight with your eyes at the lake surface. So this is a Winogradsky column if you want to know what happens and how microbes grow. And these colors are formed because of microbes. You have purple sulfur bacteria, you have green sulfur bacteria, you have cyanobacteria that make all these colors and layers from these layers in this Winogradsky column. And this is a small experiment that is being carried out in high schools of United States, specifically in Pittsburgh. And it is an initiative by Dr. Vaughn Cooper. He also is one of my supervisors under whom I am doing my postdoctoral research. So you can ask me more in details about this particular experiment if interested. But I'm going to quickly go through what this experiment is about. This experiment is a way to use microbes as model to study evolution. Now, evolution is how you evolve from your ancestors, right? But why microbes are very useful in studying questions about evolution is they grow faster. A microbial cell can divide in 20 minutes. Sometimes, depending on species, they can take a day to divide, but this is still faster than what we as organisms do. So they can grow faster. They can form large population sizes in a confined place, and therefore they are easy to handle. The best other quality of microbes is that they can be frozen and thus can be revived again. And this is something not possible. I mean, I know there are sci-fi movies that have been talking about freezing or making humans to sleep, freezing them and then waking them in 10 years. But practically it's not possible right now on this planet. But what is possible, this is possible with microbes. And so you can do these experiments, this generation of experiments with microbes. So this particular evolving stem protocol involves growing microbes. So what we do is we have this tube, we have this medium that is nutrient medium for microbes to use. You can use any medium like milk or even a laboratory defined medium. And you add microbes to it, okay? And then you have a bead. In 24 hours, microbes grow in large numbers, and they also attach to the bead. And this is the bead that we take out from the environment and introduce to a new environment. And this is called as microbial evolution model. So we introduce it to a new environment containing another different colored bead. And we go on doing this until say day 6 or so. So from day 1 to day 6, you already have like around 60 generations, like, approximately counting 6 generations per day. You have 60 generations just in a week. And just imagine this is your ancestor population, right? You can freeze it, and you can also freeze your day 6 population. And then you can revive it on a particular day and see what is the difference between these two. So what you can see is this is your ancestor, and this is how the ancestor colony would look like. Whereas your evolved colonies or evolved population looks like this. It makes different characterization. It looks different from the ancestor. And this is nothing but a diversification, something that actually happened throughout is actually happening throughout the years on this planet. You diversify into new species. Okay, so this is not a diversification into new species. When I'm talking about bacteria, this is just diversification within the species, just like what is happening in these birds. These are called as Darwin's finches. what Darwin observed is that this particular bird, when it diversified into different environments, it started using different food source. And because of the different food source it used, it developed a different kind of beak, but it remained same species. So this is called as diversification, and this is what we see in microbes. And diversification is an important step for studying evolution. And observing diversification in a week time in a classroom is a very unique concept. And I think if people are interested, they can do this in a large set. And if you have any questions, you can ask me. How you can be a microbiologist? The basic step is to do a bachelor degree in either microbiology, biotechnology, or biochemistry or genetics. And once you are in bachelor's college, you would know the next steps, whether you want to do masters, whether you want to go in which line you want to take further. What does a microbiologist do? Like, what kind of jobs you can do? You can be a research scientist, someone like me, who can work in industry as well as in academia. You can be a professor and teach, and also supervise students like me, or PhD students or master students. You can work in industry, mostly like qualitative analyst. Many food industries require people to check the quality of food, if it is contaminated or not, if it has higher shelf life or not. And people want so companies need these people to do this qualitative checks at each step of their production houses. So these food industries, cosmetic industries, pharma companies do require microbiologists. At the end, you can also work in forensics. You can work as a path lab microbiologist, studying the blood samples of the patients. At the same time, you can work as epidemiologist. What epidemiologist is, he or she is a consultant where they study how epidemics are caused, like how H1N1 virus that causes bird flu spreads from a particular location to other parts of location. And these are the studies that an epidemiologist does and gives a report to, say, a hospital or to a World Health Organization. So this is what an epidemiologist does. They study how diseases spread and become epidemics. Lastly, how you can get more information about microbes. There are very good institutes in India, two of which I know that let public come and see, or even students come as field trips or picnics in form of picnic, industrial visit, to visit them. And one is National Institute of Virology in Pune, and National Center of Biological Studies in Bangalore, NCBS and NIV. I am sure there are other research institutes in Delhi and everywhere else. You have Jain CSIR in Gujarat. I don't know much of these institutes if they allow public visits, but I'm sure they do. And if you write to them as a school principal or a school teacher, they would respond to you. That is one way. Or you can host a scientist. Maybe in school, the scientist can come and tell more about microbes and their research. If not, you can also read certain magazines. A school library can subscribe to these magazines. They are Sanctuary Asia, National Geographic, Scientific American, New Scientists. Sanctuary Asia is one of the Indian magazines. And this is something that I used to read when I was small, and that got me interested into genetics and microbiology. So this is something that school libraries can subscribe to if they want to know. Say the syllabus that you have for 11th grade students, I think something like that can be introduced, or like a small chapter can be introduced to in small grade, like 8th grade. I remember in 8th grade, we had information about adaptation to environment, and you had like plant pictures, mimosa, which is like touch me not plant, and those kind of characters of plants, like how plants behave to a stimuli. or even how our skin behaves to a stimuli. So maybe such cartoon or kind of like small processes that involve microbes can be introduced, like curdling of milk, or something like what is the structure of a mushroom, and what are the basic differences between different groups of microorganisms, can be introduced in 8th grade. Microbes, they can attach to number of surfaces. We use plastic beads because they are cost friendly. But of course, you can use glass beads. But plastic beads are easy to find, easy to produce, and they are cost friendly. This plastic is a high grade plastic, so it is not something that you can use and throw. We reuse it. So it's also eco-friendly in some case. Not very eco-friendly because it's still plastic, but yeah. So we have plastic beads, but you can also use glass beads. So there are some bacteria, I can name them. They are like Salmonella, Staph aureus. They are considered infectious. So bacterial infections are range from pimples on your skin, or skin scratches that you see, or sometimes when your wound gets infected when it's not covered properly or disinfected properly. So these are skin bacterial diseases, and you can see some things like Staph aureus in these diseases. Bacteria also form some respiratory diseases like pneumonia. They also form ear infection or urinary tract infections. They also give you dysentery. Dysentery is mostly caused by amoeba, but they can be also caused by, like, cholera is bacterial disease. Even sometimes like loose motions are salmonella disease. So these are some bacteria that I can give names of, but then these are the diseases that you know and you hear about every now and then: cholera, dysentery, loose motions, urinary tract infection, wound infections, acne, pimples. Because of this curdling of milk project, you can also introduce something like fermentation. So how you make dosa batter, that could be introduced, because that fermentation is also by yeast or microbes. Something that we do every day in day-to-day life, and also baking of cake or bread, that can be introduced. What else? Maybe people can observe the mold, which normally is discarded on if it grows on bread. I think there are very little things that you can do without a microscope. So if a school can have a microscope, then there are a lot of things that you can do. You can take samples from your teeth and see them under the microscope, what kind of bacteria there are, what kind of microbes there are, how they move, something that you can't see with your naked eye. But other than that, very limited things. But then I did discuss few things. If you are interested what grows in your contaminated tea or contaminated food, of course you can do this jelly thing and put the food drops on that jelly and give it a day or two to grow in form of a colony. Bacteria are not considered infectious at primary level. So for example, what happens is when you get common cold, it is caused by a virus, but then your immune system gets very weak, and then the bacteria that is already present as a, like body microbe, gets opportunity to cause disease when your immunity gets weakened. So bacteria are considered infectious at secondary level. I hope that answers your question. But normally for Indian immune system, I think we are exposed to so many harmful bacteria in a way that our immune system is strong. to combat them, and therefore most of the diseases you don't even see. You don't even feel it in form of fever or any other form of disease kind of feeling, because your immune system is strong enough to combat them at every instant. Also, the body microbes help immune system to combat them. Fungi and algae can be observed by magnifying glass. But if you're interested, I think there are foldable microscopes available. And I saw it on Amazon.in, but I don't know if other places sell those. And I think they are available so for rupees 500 to rupees 1000. They are foldable microscope. The only thing is I've never used them, so I don't know how effective they are, but there are many articles on them where the person who invented them was also Indian, who was in one of the American labs. She invented them, and they are now used in some schools, but I have never used them personally, so I don't know how effective they are. But I think they should be effective because there are many articles that talk about them, and that's available on Amazon.in. It's very hard to judge, right? So some bacteria are useful, and therefore you don't always wash your hands with sanitizer. So people nowadays use sanitizing agents. Overuse of sanitization is not good because it wipes away also useful bacteria from your hands. But it's a good practice to wash your hands before you eat your food, because there could be some harmful bacteria on your hands. You don't know because you touch your hands everywhere, right? You can't monitor them. There is light microscope, which is the simplest form of microscope. It has different ranges of magnification. So you can enlarge or zoom with using different lenses, just like any other camera. Then there is, so for example, viruses, you can't use a light microscope to visualize viruses. So there is this huge setup of microscope called as electron microscope that is used to visualize viruses. And electron microscope is also used to visualize certain other structures, very minute protein structures on bacterial surfaces or any other cellular surfaces. Even on our cells, sometimes cancer cells need these microscopes to visualize them properly, like how are they growing and everything. So there is electron microscope. Then there are atomic force microscope that use atomic energy in order to make structures or images. This is kind of thing that is also used in telescope to make images of some things that are very far away from you. So something that are very minute, you use atoms. You just put atoms on the samples, and that atoms create an image, and something that even telescopes use. So that is atomic force microscope. So these are few microscopes that are used. Not every microbiologist have used it. I've never used an atomic force microscope, for example, but I've used electron microscope to visualize viruses. And yeah, so as a microbiologist, if you study a particular microbe, you get chance to use these microscopes. There's so many bacterial species. Some of them are not even discovered so far. So there are many types of bacteria in air. Some bacteria that cause respiratory diseases are also present in air. But at the same time, you have lots and lots of bacteria, because bacteria are so light that they can stay in air. Like they can float in air, just like we can float in water. At the same time, you have spores present in air. Spores are the seeds from fungus. And these, therefore, when you keep your bread exposed, or if you're some food exposed, you can easily see spores germinating on them in few days. Basic structures are same. Like algae will have a chlorophyll. It will have a genetic material containing organ, which is called as nucleus. which is also present in our cells. It will have mitochondria. So these few organelles or cellular components will be same in all the algae, but then structurally they could be different. Like the way things are arranged could be different in these microorganisms. Similarly, in fungi also you have, so every fungi can emit certain pigments, right? So even in mushrooms, sometimes you see yellow mushrooms, you see white mushrooms, sometimes you see like Hitachi mushrooms are brown mushrooms. So there are like different structures, just like plants. Not all plants are same. So the way they grow non-uniformly, and so the structural difference might be present, but the essential components of cells do remain the same. Like every fungi has a stalk and a body, and it has spores, it has genetic material, it has mitochondria, even algae has mitochondria, so the essential components do remain same. Of course, and you do consume it. In form of curd, you do consume bacteria. Then there are lots of probiotics. One thing that I know is Kajol advertises for this probiotic called as Yakul. So this probiotic has bacteria, healthy bacteria that, so what happens is sometimes some people do not have healthy bacteria in their guts. So in order for that, doctors ask them to take probiotics in order to change the gut environment. And these probiotics help you like have your gut with, like fill your gut with healthy bacteria. So of course you can consume bacteria, and you do consume bacteria knowingly or unknowingly. And that's how you get infections, right? You get salmonella infections or dysentery or Vibrio cholerae by consuming contaminant water. But not all bacteria are bad. Sometimes your body helps you fight lots of harmful bacteria, and therefore you don't even know if you have any disease or not. So this is something called as microbiome, and people are studying like how microbiomes are different in different things. So your human microbiome is different than animal microbiome because of the surroundings, that how we keep ourselves. We have a bed to sleep, whereas animals have more natural environments where they are. And also they consume lots of different types of food than us. Also, there is this big study being done on people living in villages, their microbiome, versus people living in urban societies, their microbiome, how different that is. So even people living in different surroundings might have different microbiome growing on their skin, hair, gut, everywhere. So there are also different studies being done that people who have pets, they have different microorganisms, and sometimes the pet microorganism, the animal microbiome and human microbiome is shared when you have pet living in close proximity. So of course the microbiome, or what kind of composition is your bacteria, is a whole new range of study, and it's quite interesting. And you can answer a lot of lifestyle questions when you see what kind of microbiome shapes you. I'm really sorry, Akshita. I am not aware of these books right now because I have never read them. But there is this one book that I would like you to read, because it is for not for kids, but kind of for public who doesn't know anything about microbiology. It is written by Ed Yong. It is a quite popular book. It is called I Contain Multitudes. It actually talks about how microbes are part of you. You will be surprised, bacteria can live in extreme conditions. So they don't kill like a lot of, I think they will kill your gut cells more than killing bacteria. So no, what the acidic environment that we produce is harmful for our gut, but is not harmful for the bacteria so much. There are acidic bacteria that can consume the acid that start thriving afterwards. But yes, antibiotics, if you take antibiotics, they are harmful for bacteria. So bacteria, you can't know what is the lifetime of a bacteria because it keeps dividing. So one cell divides and it forms two daughter cells. And so that particular culture goes on dividing, and it goes on dividing until and unless you stop giving it nutrients. And so it could be days to months. But average lifetime of a cell to divide is either 20 minutes to a day for a microorganism. So in a day, or in 20 minutes or in a day, they can divide and form daughter cells. For example, if you see a easy bacteria like a lactic acid bacteria that grows in your curd, it can divide in 20 minutes. So what people do is when they divide, there is one part of the bacteria. So for example, just consider this as a bacterial cell. When it divides, it becomes two, right? So this is an old pole, and what is formed here is a new pole. So there are studies done on how to measure the old pole and how longer does the old pole survive. So there are these few techniques that scientists study in order to know how long does the contents from the original bacteria survive. But you can also monitor it by time-lapse microscopy, that how fast one single cell is dividing into two. There are lots of videos that you can find on YouTube about dividing bacterial cells. Just go and look at them, and they are beautiful as well.